So my name is Ralph Kittler.
I'm Angelique Whitehurst.
So my name's David McFadden.
My name's Ralph DeBaradinas.
My name is Jim Amatruda. I'm a pediatric oncologist here at UT Southwestern, and my area of interest is the genetics of childhood cancer, especially sarcomas.
I'm an assistant professor of pharmacology in the Eugene McDermott Center.
And I'm an assistant professor in the Simmons Comprehensive Cancer Center.
And my lab studies the role of altered metabolism in disease states, including cancer and genetic diseases in kids.
I'm trying to understand at the bottom what causes these cancers and how we can devise better treatments.
And my specific area of expertise is genomics, computational biology, and I'm primarily interested in studying the role of transcription factors in cancer.
And I work on reactivated immunogenic proteins, so proteins that are normally only required for reproduction and development that get turned on in cancer, and I'm trying to understand how they're contributing to tumorigenesis.
And now we're kind of doing a lot of work between chemistry and biology, so using cancer model systems to find new small molecules or drug-like compounds that block the growth of cancer cells.
So, one of the real privileges of working in this area is teamwork. And that extends all the way from the hospital, where we're inspired by the patients and the families who give generously of their time to support the research that we do and give us inspiration with their courage, to the nurses and social workers and everyone there who help us take care of these kids, all the way to the lab, where I have a fantastic group of young people working with me who are very, very dedicated. Any of them could be working less hard and earning more money in another field, but they're here because they believe in the mission. And then beyond that, we have this fantastic group of investigators that we've assembled here at UT Southwestern. So Ralph DeBaradinas, Angelique Whitehurst, Ralph Kittler, and David McFadden. And these are some of the truly most gifted people I've ever had the privilege to work with. Okay.
Yeah, we're part of the Ewing sarcoma initiative here. We're very interested in the idea that the fusion oncogene, the EWS-FLI1 oncogene, regulates metabolism in a way that allows the Ewing sarcoma cells to escape the normal cell death mechanisms so that they can grow, proliferate, and eventually form a tumor, eventually metastasize. And we believe that altered metabolism is at the root of many of these problems. Ewing sarcoma is a complicated disease, and as in any form of cancer, understanding the aberrant biology involves a multidisciplinary approach. You need good cell biologists, you need people who are experts in gene regulation, which genes get turned on and turned off, and we think there's a role for metabolism as well. So this team-based approach is important and very appealing to us, because I think it gives you the opportunity that the sum of everyone's efforts will be greater than the individual parts.
So Ewing sarcoma is a type of cancer that is driven by a transcription factor that is called EWS-FLI1. And what my lab does is we study how the transcription factors affect the regulation of genes, and that is what is disrupted in Ewing sarcoma. So this transcription factor is not expressed how it should be, and we look how that affects cell growth and the migration of cells and other processes that are related to cancer.
I really basically work on new targets, so trying to identify proteins that haven't previously been implicated in Ewing sarcoma before. And so that really dovetails nicely with some of the genomic approaches, as well as the metabolomic approaches, and also the chemical screens that are being done on the team. So it's independent, but also dependent on their studies.
When I think of Ewing sarcoma, I think it's both simple and complex. From a person who's studied adult cancers and the series of genes that are changed between a normal cell and a cancer cell, in many adult cancers, there's hundreds and hundreds of changes in each cancer. Ewing sarcoma is simple in the fact that there's not many changes between normal cells and Ewing sarcoma cells. And we know what the key change is. It's the EWS-FLI family of fusions, this chromosomal swap that happens. But it's complex because the EWS fly fusion is not straightforward. It's not a simple protein that's encoded by this fusion that we can design a quick way to block its action. It seems to turn other genes on or off and basically mess up how genes are turned on and turned off in a cell, and somehow through some unknown way that causes cancer. And so my sort of approach as I looked at this was to say, ask a basic question. What happens in a Ewing sarcoma cell when you can figure out a way to turn off EWS fly? And we can't do that in patient tumors or anything right now, so we got cell lines that are derived from patients with Ewing sarcoma. We can grow these in the lab, we can grow them in petri dishes, and then we use genetic tricks to say, let's turn off EWS fly and try to find out what happens. So we've toyed around with several different methods in the past year and a half, and we came upon a really cool technique that basically creates a self-destruct button in a protein where we can control it in the lab. We can say, turn off this protein, and it'll self-destruct. So we engineered the self-destruct kind of button basically into the EWS fly fusion gene, and we've just been able to do that. And now in the next several weeks, we'll have an answer, I think, to what happens in a Ewing sarcoma cell in a petri dish when you push the self-destruct button and EWS fly self-destructs.
I knew for quite a while I wanted to be a cancer doctor. I never knew I wanted to be a fish farmer, but I think it's an example that we do what we need to do to tackle the problems we want to take on. In our case, we chose the zebrafish because it actually is a very, very powerful model system that we can use in the laboratory to take on big questions. Where does Ewing sarcoma come from? What sort of cell does it come from? How does this one protein, EWS fly, that we all know way too much about now in this community, how does that actually turn a normal cell into a cancer cell? Why does Ewing sarcoma happen in kids and not so much in adults? All of these questions, plus more, looking for new treatments, we can use the zebrafish to study these questions. And again, not only one system would work. We need fish, we need mice, we need cells, we need chemicals, we need computation. But it's something that we can bring to the table. We have a number of years experience, and actually one of the most exciting developments is we were able to show that the same gene that causes Ewing sarcoma in a person causes the same cancer in a fish. Once we saw that, we were off to the races, and we've been working very intensively to use that system to try to learn more about the disease and most importantly to develop new treatments. Once you become part of this pediatric cancer community, you realize what a sense of mission we all share. We absolutely have been fortunate to receive funding from the federal government, so that's everybody's tax dollars. Thank you, and we take that obligation very seriously. But it's a whole other level when it comes from groups like 1 Million for Anna and other pediatric cancer charities, where people have reached into their pockets and pulled together. And that's something that inspires us and makes us want to work that much harder.